Okay, let's continue on muscle tissue and look at the steps in excitation, contraction, coupling. Okay, so the action potential is spread out over the surface of the muscle cell, as you mentioned, stimulated by the motor endplate potential, and enter the T tubules. As I mentioned, action potential is spread out over the sarcoplasmic, over the sarcolemma. They go down into the T tubules. So it brings the excitation down into the sarcoplasm. Voltage-gated ion channels are opened, which open calcium channels in the terminal cisternae of the sarcoplasmic reticulum. Calcium rushes out into the sarcoplasm, and then calcium binds to troponin. This binding of calcium to troponin moves tropomyosin out of the way of the actin binding sites for myosin. So remember, on actin, there are myosin binding sites. Tropomyosin is blocking them. Binding of the calcium to troponin moves the tropomyosin out of the way, and as soon as that happens, the myosin head attaches to the actin. So release of neurotransmitter from synaptic vesicles and subsequent binding of ACH to HGH receptors. Sarcolemma T tubules, HH binding triggers propagation of an action potential along the sarcolemma, and T tubules to sarcoplasmic reticulum, stimulate to release calcium. Calcium binding to troponin triggers sliding of thin filaments past thick filaments of sarcomeres. Sarcomeres shorten, causing muscle contraction. So we're going to see that. I think this should be in the next slide. So when the binding site for myosin on actin is exposed by myosin being moved out of the way, as I said, the myosin head attaches to the actin, and then we get what's called a power stroke. Initially it's 90 degrees and it goes to like 45. That's called a power stroke, and this moves the thin and thick filament past one another a little bit. Okay, and then after the power stroke. So the power stroke is after the cross bridge. Well, the myosin head is often called a cross bridge. Attaches to actin, it switches from 90 to like 45 degrees, and then ATP comes along, binds to the myosin head to detach it from actin. Then ATP is hydrolyzed. There's an ATPase. Myosin head has an ATPase in it, which hydrolyzes ATP. In other words, breaks down ATP to ADP and PI and releases energy and recocks the myosin cross bridge back to 90 degrees. When it does that, it's went across from another actin binding site farther down the thin filament and immediately binds, goes to the power stroke. And then ATP binds, detaches it, ATP is hydrolyzed, binds again, like that. So, so what happens is it's a ratcheting mechanism. Binds, power stroke, detach, recock, like this, a ratcheting mechanism. And this causes the thin and thin filament to slide past each other. And this is hence the term sliding filament theory. So, and this is called cross bridge recycling. So calcium binds to troponin in muscle thin filaments, causing a conformational change in troponin. Troponin changes shape, and the entire tropomyosin complex is moved. Thus, tropomyosin no longer covers the myosin binding sites on actin. Cross bridge formation. Myosin heads, which are in a cock position, in a relaxed muscle, they're already in a cock position. They attach to the myosin binding sites on actin, and then we have the power stroke. The myosin head swivels toward the center of the sarcomere, pulling the attached thin filament. The motion is called a power stroke. ADP and PI are released during this process. Then ATP binds, 3D here, release of myosin head. ATP binds to the ATP binding site on the myosin head, which causes the release of the myosin head from the binding site on actin. Then the myosin head has to be reset. So then the ATP is split into ATP and PI by myosin ATPase to provide the energy to reset the myosin head back up to 90 degrees. Now, again, once it does that, it's read across from another myosin binding site and actin farther down, immediately binds, goes to power stroke. So the important thing is, what's the role of ATP in muscle contraction? It's not the power stroke. The role of ATP is to detach myosin from actin, and then when it's split, to recock the myosin cross bridge. And then it attaches and goes to the cross and goes to the power stroke. So that's crucial. That's the role of ATP in, and this will keep on going. And so this recycling of attaching, going to power stroke, detaching, recocking is called the, and this goes around and around, and all of these gazillions of cross bridge recycling causes the sarcomere to shorten and the filaments to slide by one another. Okay, there's a sarcomere shortening. Here is a relaxed sarcomere up here, where we have an H zone, and here's a slide of this. So you can see the light area is I bands, the dark area is A bands. And then here's a contracted muscle. These the filaments have slid past one another and have obliterated the H zone. There is no more H zone. And there's very little I bands either. I bands have been obliterated in the H zone, and now we just have A bands. So it looks just dark. It's not striated anymore. And then after we turn off the action potentials, remember the tropomyosin slides back over the myosin binding sites and actin. The calcium pumps are activating the sarcoplasmic reticulum. The calcium comes off the troponin, and So first thing is the calcium pumps. This is probably the next slide. Relaxation. Nerve signals stop arriving at neuromuscular junction. Acetylcholinesterase breaks down acetylcholine in the synaptic cleft. Active transport begins in the sarcoplasmic reticulum to resequester calcium by binding to a protein called calsequestrin inside the sarcoplasmic reticulum. Calcium dissociates from troponin. This causes tropomyosin to go back over the myosin binding sites in actin and block them. And then the elastic filaments pull the thin filaments back, and the I bands reform and the H band reforms. And then we have a relaxed sarcomere. Okay. Muscle metabolism. We got to talk about where the ATP comes from. There's aerobic and anaerobic ways of generating ATP. And there's quick sources and slower sources. So ATP sources, immediate sources. One would be actually aerobic, because myoglobin. As soon as you start moving your muscles really fast, the oxygen tension goes way down. This allows the oxygen to come off of myoglobin, and you can use oxygen for a little bit of aerobic metabolism. But that's really, that only lasts a very short time. Then anaerobic respiration takes out the phosphagen system, the creatine phosphate, where ATP is made really quickly by breaking down creatine phosphate. Then you have a myokinase, which is another quick way of making ATP. Short-term anaerobic, the glycogen lactic acid. So if you need to contract your muscles really quickly, you break down glycogen to glucose, You make 2 ATPs per round of glycolysis, and then lactic acid builds up. Okay? And this is for using muscles really quickly. Long term, you want to use your muscles more slowly in a longer term, because the short term leads to muscle fatigue. Long term is aerobic, because it takes time to get the oxygen to muscles. And so you have to contract your muscles slow enough in order to get oxygen to them, then you can stay aerobic, and that doesn't cause muscles to fatigue nearly as quickly. So here's the ATPase breaking ATP down to ADP and PI. Here's myokinase can take 2 ADPs and make an ATP from them and generate an AMP, an ATP and an AMP. And then creatine kinase here can take ADP plus creatine kinase and make ATP and creatine. And these are all really fast ways of making ATP. But they only but they're exhausted really quick. Within the first few seconds of muscle contraction, they're used up. Well, here we need to go over cellular respiration a little bit in order to understand this this muscle metabolism. Remember, glycolysis to pyruvate is a anaerobic pathway. There's no oxygen needed for that. It only makes 2 ATPs, but it does so really quickly, right? So the pyruvate is the branch point. So pyruvate is made, and there's enough oxygen in the muscle cell, then the pyruvate is converted to acetyl-CoA, and it'll go into citric acid cycle, and you'll make NADHs and FADHs, and you'll send those to electron transport, and you'll make lots and lots of ATP. And then the used up electrons in electron transport are given to oxygen, and then water is made. But if the oxygen concentration in the muscle is really low, then pyruvate is converted to lactic acid, and you keep on going through glycolysis. And you don't even go into mitochondria, right? But as I said, lactic acid builds up. It's one of the major reasons for muscle fatigue. It has to be reconverted to pyruvate, and eventually you have to go in the aerobic direction. That's why you have this oxygen debt at the when you're really exercising really rapidly. You get this oxygen debt, and after you stop, you have to breathe heavily, because you got to repay that. You got to rebuild up all that creatine and phosphate and all the stuff that you used up. So lactic acid has to be reconverted to pyruvate, and you eventually have to go to aerobic group, because we are obligate aerobic organisms. We have to use oxygen eventually. We can temporarily not use oxygen, but then we eventually have to stop and use oxygen. So short-term high-intensity exercise requires anaerobic methods of making ATP from carbohydrates. You have to use carbohydrates. In order to do anaerobic means, you got to use carbohydrates. You can't use proteins and fats anaerobically, just carbohydrates. Long-term, low-intensity uses aerobic methods and can use carbohydrates and fats and proteins. And so the two different types of exercise, resistance exercise, which means running real fast or lifting weights, for example, uses anaerobic means, and they build bigger muscles. But cardiovascular, cardiorespiratory exercise, cardio, doesn't make big muscles. You don't need a bigger muscle. You build an oxygen delivery system is what you do. It improves oxygen delivery system so someone can maintain high intensity exercise for a longer period of time. So you're a world-class marathon runner. You're running sub 5-minute miles for 26 miles. I mean, that's pretty impressive. And so what you're doing is actually making blood vessels, and it's called angiogenesis. Now, smooth muscle, you can make new muscle cells in smooth muscle. And that's what you do when you do cardio. You make more blood vessels to bring more blood more rapidly to leg muscles, for example, so they can go faster and faster and still stay aerobic, so they don't fatigue as quickly. Because remember, if you're lifting those weights and doing it rapidly, and all of a sudden there's just one, you just can't get the next rep because your muscles are shot. There's too much lactic acid, too little ATP, and you got to put the weight down. But if you're jogging, how long? So you only do that for a little while. If you're jogging, how long can you jog? You can probably, you can jog for a really long time because you're going slow enough to get oxygen to your muscles, and so it's not going to fatigue as much. Okay, so here, phosphagen system, immediate energy source in the first 5 seconds. You're starting to run. In the first 50 meters, 5-6 seconds, you've blown through the phosphagen. Next is the anaerobic right here, 400 meters, maybe go once around, 50-60 seconds, then that's used up. And then the yellow is aerobic. If you're going to go long farther than that, you have to turn to aerobic mechanisms. So the oxygen debt, replacing body oxygen reserve, replenishing the phosphagen system, oxidating the lactic acid, and you have an elevated metabolic rate. So you're using a lot more oxygen after you've done this exercise. So even if you do an aerobic exercise, you still got this oxygen debt, and that's why you're breathing heavy after exercise. Okay, types of contraction. Power related to diameter of fiber. Bigger diameter fiber, more power that can be generated. Speed, what variant of myosin ATP present? So what we have is fast twitch and slow twitch myosin ATPases. So fast twitch is really rapid muscle contraction, and this uses anaerobic metabolism and glycogen and glycolysis, and then slow twitch. And so there's not very many mitochondria in those fibers. But slow twitch is using aerobic mechanisms. They contract more slowly. They use oxygen, so they have lots of mitochondria. So we have oxidative fibers. Lots of mitochondria in myoglobin, and they're red. And they are they really work best with aerobic metabolism, so they're slower twitch. Glycolytic fibers, few mitochondria, little myoglobin, lots of glycogen, and this is the white fibers. And so those are the ones that you use really quickly. That can generate really a lot of tension really quickly. Okay. So classes of muscle fibers. Slow oxidative. Slow and less powerful contractions, they're very resistant to fatigue. So your back muscles, remember they're like contracted practically all day long, right? To keep you erect. But they're slow and they're not that powerful, but they just have to hold you up, and very resistant to fatigue. Fast oxidative, they're intermediate. They're fast, powerful, mostly aerobic, less myoglobin than the slow oxidative. And that's leg muscles. So a lot of your leg muscles are these fast oxidative. And then fast glycolytic, large, most prevalent, power and speed. Little myoglobin, anaerobic primarily, and they're very fatigable. And this is in the arms. So I noticed that in the gym, you go onto a stationary bike and you start riding a stationary bike, and you can go pretty fast for quite a while before your legs really get tired, okay? And these are fast oxidative because even though you're going faster, you're still staying oxidative and using oxygen. and you can go farther before they get fatigued. But then you go to the arm bicycle. There's a instrument in the gym where you move your arms like this rather than your feet. And I do that for like 5 minutes and my arms are like dead. They're dead because you're using anaerobic metabolism there, and that's very fatigable. Those are fast glycolytic fibers and they're very fatigable. Now you can change this a little bit by training, okay? And you might know if you were on a track team or did any kind of sports, what you were better at. Whether you're better at, a sprint, doing a 100-meter dash, or were you better at the longer distances, like the mile or 2 miles, something like this. And it all and actually it could be genetically, it's kind of genetically determined. If you've got a lot of if you got more than the usual fast glycolytic fibers in your legs, you're going to be a really fast runner, okay? And not so good in long distance. If you have a lot more fast oxidative in your legs than the usual person, you're going to be a really good long distance runner. So, but you can change that to a certain extent by training, but the person who genetically has more fast glycolytic fibers is always going to be a little faster than you. If you both have the same amount of training, but this guy has genetically more glycolytic fibers, he's going to run faster than you no matter what. So like Usain Bolt, this guy is, the fastest man in the world, and he was genetically blessed with a lot of really fast glycolytic fibers, versus who's a good marathon? Forget who the or best American marathon runner. Well, these guys, or the Ethiopians, who like win practically every marathon, they have genetically more of the fast oxidative, and they can go long distances very rapidly without and still staying aerobic and not getting fatigued. So most muscles contain all three types. Some are more depending on function. Muscles of eye and hand, mostly fast glycolytic. In the back, slow oxidative. Variations between individuals mostly depend upon genetics, as I just mentioned, and less so by training. Okay, then we're going to get into twitches and what a muscle twitch is, and these parts of a muscle twitch: threshold, latent period, contraction phase, and relaxation phase. I'm going to show you that in a later slide. Changes in stimulus intensity. So motor unit recruitment and all or none law. The all or none law means if this muscle cell is stimulated to contract by its motor neuron, it does so 100% or nothing. It's all or none. So motor unit recruitment is that if you have to pick up something, something that has a particular weight, you have to recruit enough motor units to generate enough tension to pick it up. If it's heavier, you have to recruit more motor units in order to pick it up. Or if you put something on top of it, it becomes heavier. So the tension generated by a muscle is directly related to how many motor units have been recruited, how many motor units are being utilized. And this is we used to actually do this in A&P, but it meant killing frogs, and I never really cared for it, so we don't do it anymore. We don't kill any more frogs. As you set up a muscle in this arrangement like this, And this is it's a force transducer. So we have this muscle hanging there, and when it contracts, it pulls up on this arm, and it's sent to a chart recorder or a computer, and you can get this muscle twitch. And then you can turn on and what you have an electrode stuck in the muscle, and you have a stimulator where you can determine how much voltage you're going to use to stimulate the muscle and how often you stimulate it. So and then you have a switch on the stimulator. So you push the button on a stimulator, and a stimulus goes to the muscle. And there's the stimulus right there. There's a latent period where you don't see any kind of contraction. This is the muscle tension here, which is force. And there isn't any force in that latent period because it takes time for the action potential to travel down the T tubules to open up the calcium ion channels for the calcium to come in, bind it to its troponin, moves it to a myosin out of the way. And then you can get a contraction period and force generation. And then a relaxation period, and it goes back to normal. So that's a muscle twitch. Muscle tension or force on the y-axis and time on the x-axis. In here is increasing voltage. So you have a voltage stuck in the muscle, and here is subthreshold voltage. You're giving it a muscle a stimulus, but not enough to to cause any exponentials to be generated in any of the muscle cells. So you don't get any tension generation. Finally, right about here, you give it enough so the nearby motor cells have been stimulated and they fire and they generate a little bit of tension. You turn up the voltage, turn it up, and then you start recruiting more and more and more muscle cells to firing. And so the tension starts increasing the more you turn up the voltage until you get over here to get maximal contractions. At this voltage level, you are now contracting every single muscle cell in the whole muscle, and therefore you increase the voltage, you're not going to get any more tension because you're already contracting every muscle cell in the muscle. And then so then you can start changing the frequency. So we turn up the voltage and we got that effect. What happens if we turn up the frequency? Then you have twitches can summate. In other words, if you get this muscle twitch and you wait and then you stimulate it again, the same voltage, and you get another twitch at the same size. Right? But if you stimulate the twitch before it relaxes, these twitches can add together and you can get more muscle tension. So twitches summate. TREP is what's called incomplete. TREP is these, you're stimulating fast enough where you're not letting it relax totally, and they're building up into stronger and stronger muscle twitches. Incomplete tetany is even more than TREP and making more tension. And tetany is maximal sustained contraction. And that's not really seen in the body. That's an artificial thing. Sustained contraction is body achieved by contracting different motor units in an overlapping pattern. So you want to have a sustained muscle contraction. We already talked about this earlier. If you want to keep on holding this thing up, you're utilizing several motor units to do this. And they're gonna get fatigued. So it's an automatic pattern with those muscle units that if you want to keep on sustaining contraction, you turn off those motor units, turn on other motor units to take over in a revolving pattern to keep on holding that thing up. Okay. An amount of tension generated is related to a couple of things. The amount of calcium present, how much calcium comes in. The more calcium comes in, the stronger the contraction. That's true for a cardiac muscle as well. And the length of muscle fiber. We're going to look at what's called a length tension relationship. uh, skeletal muscles generate maximal tension at a particular length. And if it's shorter than that, they generate less tension. And if it's longer than that, they generate less tension. So here is what you get if you turn up stimulus frequency. So here are muscle twitches that you stimulate. Here you get a twitch, you let it relax completely, you do it again, you get all the same size twitches. Here you turned up the stimulus. So now you're not letting the muscle relax completely. And that's called trep. And you get and they summate together and you get larger and larger and larger contractions each time you stimulate, because you're not letting the muscle relax. And then if you do it even farther, you get what's called incomplete tetany. And then up here is your stimulating so rapidly. Now here in incomplete tetany, you're letting it relax a little, you let these fibers relax a little bit. But in tetany, you're stimulating it so rapidly that they cannot relax at all. So they stay completely maximally contracted. And that's called tetany. Okay, so contraction strength can also be related to temperature. A warmer muscle can generate more tension than a cold muscle. Weaker at a lower pH. So this is why one of these muscles get weaker and you get fatigued, because you generate a lot of acids and your lactic acid, and so the pH of your muscles start to decrease, become more acidic, and the tension generated by the muscle gets less. State of hydration. Dehydrated muscle doesn't work as well, generate as much tension as well-hydrated muscle. And remember, stimulus intensity is recruitment of motor units. How many motor units? If you get a big stimulus, if you get a small stimulus, you'll recruit a few motor units. A bigger stimulus, you recruit more motor units, and you get more tension generation. And then stimulus frequency, TREP, temporal summation. You are stimulating the muscle rapidly enough so that the muscle fiber cannot relax completely, and you get higher tension generation. Okay, muscle tone. Involuntary contractions of random motor units when the muscle is relaxed. So when the muscle is relaxed, and this comes from the cerebellum. This is called muscle tone. There's a program coming from your cerebellum, going to your muscles and contracting muscle cells in a random way. And it's not enough to cause a noticeable macroscopic contraction, but so these contractions don't move the muscle, but it keeps the muscle ready for instant action, and it also helps keep the muscles healthy. There's almost a trophic relationship between stimulating a muscle and it being healthy. This is why muscles atrophy if you don't use them. You use them or you lose them. So it has this trophic function, and also these muscle tone stretches does what it means, stretches the series elastic element, because a muscle works like you have a spring attached to a brick, and you want to pick up the brick by pulling on the spring. So you pull up on the spring, and what happens? The brick doesn't move. You have to stretch the spring entirely out before the brick moves. Well, that's the same with a muscle. There is a stretchiness to the muscle, and you have to stretch the elastic elements of the muscle before you cause a contraction. So muscle tone will keep that series elastic element, keep the muscle stretched to the right level so that's instantly usable. Okay, isometric and isotonic contractions. Isometric, same length. Metric means length. Iso means the same. So isometric is same length, changing tension. Isotonic, changing length, same tension. So here's the thing. if in when you pick up something, pick up like this, you have to generate enough tension for me to lift this thing. Right? And in the beginning, it's isometric because I generate a little bit of tension and then I generate enough tension to cause it to move up. So this is isotonic because it's same tension. I have to generate a certain amount of tension to lift this thing, right? And once I generate enough tension, then it stays the same. In the very beginning, it's sort of isometric because you haven't generated enough tension to pick it up yet. But once you've done, it becomes isotonic. And so you have changed, and the length of the muscle is shortening, and but the tension is remaining the same because I only need a certain amount of tension to lift it. Sorry. Okay, well, what if I want to lift this table over here? It's too heavy for me to lift. I can try to lift it, and I can generate more and more and more tension. So tension is changing, but the length is not changing because I'm not lifting it. So isometric is trying to push against something or lift something that's too heavy to lift. So the tension is changing, but the muscle is not shortening. And both will stimulate and develop muscles, both isometric and isotonic exercises. So length-tension relationship. Muscle cells generate optimal tension, I already mentioned this, at a particular length where there is enough overlap between thin and thick filaments, but not too much. And there is the strength-tension relationship. Muscle tension on the y-axis, sarcomere length on the x-axis. So here, there's optimal overlap to generate maximal tension. Here, there's less overlap, so there's less space. So you have a nice big H space right here, so you have enough room to contract, but if you have less, if they overlapped a little bit too much, then there's less room for them to contract. I mean, the ends of the myosin filaments are closer to the Z line than they were before, so they can't contract as much. If they're overstretched, then there's not as many myosin cross bridges attached to the actin as normally because it's overstretched. So the fewer number of cross bridges attaching means fewer tension generated. So if it's understretched or overstretched, you generate less tension. So in your muscles are stretched naturally in your body to that resting length, to that length where you can generate the most tension. Okay, fatigue. As I mentioned before, decrease in glycogen stores, not enough calcium at the muscle junction or in the SR, sodium potassium imbalances, increased phosphate ion buildup, lactic acid buildup. So all these will lead to muscle fatigue. Okay, so resistance versus endurance exercise. As I mentioned, resistance leads to muscle hypertrophy and maybe a little bit of hyperplasia, but mostly hypertrophy, just bigger muscle cells, bigger, stronger muscle cells. Endurance leads to angiogenesis, cardio, better oxygen delivery system. And you really, each time you do that, you actually damage the muscle a little bit. But then, and that's why if you work these muscles really hard this day, work different muscles the next day, or give it a little rest. So it'll fix itself and make it a little better than it was. So muscles, you use it or you lose it. Muscles will atrophy from lack of use. And some effects of extreme atrophy could be irreversible. That muscle will not come back to what it was. Effective aging. sarcopenia, loss of muscle mass of legs, natural, not due to a great decrease in physical activity. It's a natural, so that's why it's good to be exercising your whole life to try to limit the extent of sarcopenia. Like I said, it can be minimized by continued physical activity. And then fibrosis is replacing muscle tissue by fibrous connective tissue, and of course that doesn't contract. Okay, cross-training and conditioning. So cross-training is using both aerobic and anaerobic exercise. So you're jogging and you're staying aerobic, and then you start running really fast for a short period of time. It's called a fartlek. And then you stop, and then you go back to running more slowly. So that's called cross-training. And deconditioning is, again, atrophy of muscles that are not used. Okay, that's it for skeletal muscle. The next part is cardiac muscles. So we're going to hang on, talk about that in the next video. So this will be 203 muscle 2. So I'll see you later.